Parkinson's disease is a neurodegenerative disorder that affects dopamine-producing neurons in the brain, and it develops slowly over several years with a diverse set of symptoms. The severity of Parkinson's disease varies from person to person, and symptoms include tremors, which usually begin in a limb, often in your hands or fingers also, and bradykinesia, which is slowed movements, and it can make simple tasks very difficult and time-consuming. And it can also cause speech impediments. And the cause of Parkinson's disease is currently unknown, but scientists believe that genetics and environmental triggers have a large impact on influencing Parkinson's disease.
So for membrane trafficking, we're going to be taking a look at a dopaminergic neuron, and specifically the presynaptic terminal. So what we have here in this figure on the left is a physiological dopaminergic neuron, whereby the neurotransmitting vesicle undergoes snare-mediated vesicle docking to release the dopamine into the synapse. And then we can also see in that figure that the autophagosome is capable of leaving the synapse and preventing the protein accumulation, specifically alpha-synuclein. And in the figure on the right, we can see this is the presynaptic terminal in a Parkinson's disease patient. So what we have here is a large accumulation and formation of these oligomeric alpha-synucleins, and these take up a lot of space in the presynaptic terminal and actually prevent the overall number of secretory vesicles that can be formed, and ultimately that leads to reduced dopamine release overall as well. But not only does it do that, but the accumulation of the alpha-synuclein also impairs the autophagosome from leaving the synapse to fuse with lysosomes, and obviously that's going to prevent protein breakdown and thus more accumulation, so compounding over time. So we're going to be taking a look at leucine-rich repeat kinase 2, or LRRK2, which is a protein, and it's significant towards Parkinson's disease because a mutation in its gene is the most common cause of autosomally inherited PD. What this mutation is a missense mutation of a glycine to a serine at position 2019. And the exact function of the LRRK2 protein and how this mutation actually induces Parkinson's disease are both still unknown and still being researched. But we found a couple proposed mechanisms that we thought were interesting. So the first one was that this mutation causes the LRRK2 protein to become hyperactive. And when it becomes hyperactive, it's kinase, it's going to be over-phosphorylating. And in this case, we're going to be taking a look at the MAPK-ERK pathway, which is just a protein communication line from the cell membrane to the nucleus. And over-phosphorylation of that pathway, they saw increased autophagy, and that increased autophagy, what it's going to do to neurite or neurocommunication is actually reduce it. So it's going to have neurons are going to start having reduced axonal and dendritic length, which affects overall neurocommunication. And the second pathway or mechanism we saw was involved endolysosomal dysfunction, which was a result of Rab7-dependent paranuclear lysosome clustering. And endolysosomal dysfunction is consistent with Parkinson's disease in the sense that we have impaired protein and cell cleanup.
Alpha-synuclein is an abundant brain protein, normally functioning in neurotransmission and trafficking lipid vesicles in the brain. Duplication and triplication of the asynuclein gene increases the risk of early onset Parkinson's disease. Parkinson's is characterized by alpha-synuclein protein misfolding, where it becomes distorted and then malfunctions. This malfunction can lead to phosphatidylglycerol, vesicle disruption, and asymmetric membrane deformation. This may affect transporters, vesicle release, and neuronal apoptotic response, as well as inhibiting macroautophagy and chaperone-mediated autophagy. The folding malfunction leads to the accumulation of oligomers and amyloid fibrils, which are the main content of Lewy bodies. These clusters are toxic to dopamine-producing cells, and is this drop in dopamine that triggers many of the symptoms of Parkinson's disease. Alpha-synuclein preferentially binds to mitochondrial membranes rather than synaptic-like vesicles, and prefers low lipid density membranes with irregularities. The protein inserts itself deeply into the membrane. Previously, it was thought to lie flat upon the membrane surface, and it makes membranes less rigid and more porous. Multiple protein mutations linked to Parkinson's pathogenesis cluster in distinct parts of the endolysosomal system, such as clathrin-mediated endocytosis, lysosomes, and retromer trafficking. ATP13A2 is a gene that encodes an endolysosomal protein associated with the lysosome specifically. Loss of this gene is shown to cause lysosomal abnormalities, protein aggregation, and motor deficits, all seen in Parkinson's disease.